Assalamualaikum, class 10th. So this is your biology class, and the topic we are going to study today is related to Mendel's law of inheritance. So actually Gregor Mendel was a monk. He was a priest in Austria. He developed the fundamental principles of genetics. Mendel proposed that there are special factors in the organisms which control the expression of traits. Actually, he was hinting towards the concept of genes. Um, but definitely at that time it was unknown. These factors were eventually termed as gene. Mendel selected pea plant Pisum sativum to carry out a large number of experiments, and in his writings he gave reasons for the selection. Why did he select Pisum sativum? So these are the reasons, uh, and these are the actually the features which a seed should contain, and any organism that should contain for genetic experiments. Okay, so it says that when we are experimenting about genetics, we must be having these features. There should be a number of different traits that can be studied. So there should be a different number of traits that can be so only one trait, for example color of, or if you are studying about an organism. For example, you are studying about a specific species of a mouse and it has just white color. So white color and black color. So what would be the result of your experiments? It would be related to only one trait, and that is color. So it can have different sizes, it can have different eye color, so many different things are there. It can have different fur texture and many different things. Okay, so the organism should have contrasting traits, which means that the trait of height, for the trait of height there should be only two very different phenotypes, tallness and dwarfness. So if there is something in between, you will get confused. If there is an average height, if there is a medium height, you will be confused about the input. So there should be two contrasting traits, for example colors, yellow color or green color. For example, there should be nothing in between. There should be, like, for example, 0 or 100, nothing in between. The organism, if it is a plant, should be self-fertilizing, but cross-fertilization should also be possible. So if we are studying about plants and our specimen is a plant, so it should be self-fertilizing. Why? Because self-fertilization is easy, but cross-fertilization must be there to study about the contrast in the result of traits. The organism should have a short but a fast life cycle. Why? Because we need to study the organisms and we need to get towards the result easily. So whenever we want the results easily, and we want our um, research to be to have a shorter span of time, it should be concluded easily. So for that we need a fast life cycle. These features were present in pea plant, and normally, so according to Mendel, the Pisum sativum, which was the pea plant, was very um, was very perfect um, for these experiments. Why? Because it was fulfilling all the different requirements, and all of the different features were there, because it has two different colors, distinct features. The life cycle was easy, and he selected the right organism. Cross pollination would also had been done. Okay, and the second thing is, it was a self-pollinated flower. So self-pollination was also there, so that is why he selected that. Um, so this was the reason why he selected Pisum sativum. Mendel's law of segregation. Now Mendel studied the inheritance of seed shape first. For this purpose, he crossed, reproduced. He crossed. The word crossed would be used, and crossed means reproduced. Two plants having one contrasting trait, and that was seed shape. And whenever you are going to study one trait means one quality, whether it could be color, whether it is size, anything. We say, when we are talking about one trait, one quality, one feature, we say that this is a monohybrid cross. Okay, Mendel crossed a true breeding round seeded plant with a true breeding wrinkled seed plant. So the seed was wrinkled and round. So what were the results? As all resulting seeds of the next generation were round. Okay, here in this diagram they are showing about the we were talking about the cross between round and wrinkle shaped pea seeds, but here they have shown. First we have to go through this. These were the seed shape, two seed shapes, round and wrinkled, two seed colors, yellow and green. So contrasting traits were seen and many traits were also present. Yellow and white and purple flowers. So seed shape was flat and one was flat and the other was constricted. Again, pod color was different, yellow and green, and the flower position was also different. It is axial and that is terminal. Terminal means at the end and axial means in between. And the stem length was also different, long and short. So that is why he chose Pisum sativum, because many different traits were present in different species. All resulting seeds of next generation were round. So see here, actually what he did was, parental generation is this parental generation and this is the filial generation, F1 generation. Okay, F1 is the first filial generation and then F2 is the second filial generation. Like first filial generation means the resultant offspring, first resultant offspring. So when he crossed round and wrinkled, what was the result? All the seeds were round, and then what he did, now see, all resulting seeds of the next generation were round. Mendel declared that the trait round seed was dominant while the wrinkled seed was actually recessive. The following year, Mendel planted these seeds and allowed the new plants to self-fertilize. Now, what happened? Self-pollination took place, and as a result, he got 7,324 seeds, 5,474 round, and 1850 wrinkled. Again, see, the result he got was round seeds were more and wrinkled were less. So the ratio became 3 round and 1 wrinkle ratio. The parental generation is known as P1 generation. The offspring is of the offspring of P1 and F1 generation. The offspring of P1 generation is known as F1 generation. Understand? And first, that is known as first filial, which has already been told you, which are already been told. The cross in the F1 generation produces F2 generation, and this is filial F2 generation and second filial generation. Okay, so see, what he did was, uh, then the resultant, see, this was 3 ratio 1, 3 ratio 1. He crossed here two round um, two round seeds, but the result he got was, see, 75% down and 25% wrinkled. Why? Because of the genotype. We can't see the genes. The wrinkle was present somewhere because of this. Because of this parental generation, the wrinkle gene was present somewhere, and that is why we got the result. And 25 wrinkled seeds were present. Why less? Because that is a recessive trait. Recessive is always less in number. Now, inherited. Now, similarly, when true breeding tall plants were crossed with true breeding short plants, all offsprings F1 were tall plants. Okay, again he crossed two breeding tall plants and two breeding short plants. Then what happened? F1, all the tall plants were there and there was no short plant. Uh, then he then he took the F1 filial generation, and then again they were self-fertilized, and then we got the ratio of, again the ratio was 3 ratio 1. The trait was different but the ratio was same, 75% to 25%. Now, Mendel concluded that the traits under study were controlled by separable discrete, means separate factors, discrete factors or genes. In each organism, the genes are present in pairs. During gamete formation, the genes of each pair, actually this is what we know about it, about inheritance now, and about the reproduction now, that gametes actually disaggregate from each other, and then they join, and then there is a union of gametes, and because of this union the resulting offsprings get the genes from the parents in pairs. So that is why we say one is dominant, one is recessive. The dominant might be coming from the from one parent, and the recessive is also coming from the other parent. From one parent dominant and from one pair recessive. It can be dominant from both the parents and it can be recessive from both the parents. Okay, these were the conclusions. These conclusions were known as law of segregation. So whenever they ask you about law of segregation, you start from here, in that during gamete formation, this, when the gametes, no, from here. So it starts from here. When the gametes of male and female parents unite, the resultant offsprings again the resultant offspring gets the genes in pairs. These conclusions were called the law of segregation. Why segregate? Segregate means segregate means separated. Understand? Segregate means something which is separated from the other. That is why he said that the genes were separated from each other and then the union happened. That is why he called it the law of segregation, because at some point of time in the parent, before fertilization, the genes were separated and only one gene was left. You always know that there are pairs of gene. We also have pairs of gene, but before fertilization the pairs of genes were separated, and then what happened when they united, the genes came from both the parents and the resultant offsprings were having different genes. Okay, and this was known as law of segregation.